In this talk, I will present the races of the stem rust pathogen in Ethiopia in the 2014-2021 period. Wheat stem rust is considered a re-emerging disease, and it is well exemplified by the occurrence and spread of races in the UG99 group. Up to date, 15 variants of this group has been identified with a geographic distribution that covers 14 countries in East and South Africa and the Middle East. In addition, recent epidemics and outbreaks, like the one observed in North Kazakhstan and West Siberia, southern Italy, and Tunisia, are a strong indication that this disease has re-emerged, posing a significant threat to wheat production. One of the most recent and devastating epidemics occurred in south-central Ethiopia in 2013 and 2014. This epidemic affected 27 districts in the major wheat-growing regions of Bale and Arsi, where about 100,000 hectares were planted to wheat. Yield losses up to 90% were reported in Digalu, a widely grown cultivar with yellow rust resistance and high yield. Digalu carries SRTMP, a gene effective to TTKSK, at that time the predominant race in Ethiopia. Through phenotypic and genotypic evaluations, we confirmed that the race responsible for this epidemic, TKTTF, was not related to the UG99 race group. Race TKTTF is highly virulent on SRTMP, as you can see in this picture, the reason why it was so devastating on Digalu, and avirulent on SR31, a distinctive phenotypic characteristic of races in the UG99 group. It also formed a genetic lineage defined as clade 4 that is different from the UG99 race group, or clade 1, as you can see in the phylogenetic tree. Two additional clades were defined, clade 2, which includes race JRCQC, and clade 3 that includes races TRTTF and RRTTF. After the 2013 epidemic, significant efforts were put to continue monitoring the disease in Ethiopia, with a strong collaboration that includes national and international partners. For today's talks, I will present what happened with the stem rust pathogen, PGT from now on, in Ethiopia in the 8 years that followed the epidemic. As you can see in this table, about 600 stem rust samples were received between 2014 and 2021, and 678 isolates were derived and race type. Samples were collected from main wheat-growing regions in Ethiopia, not only from farmer fields, but also from research plots. Although the majority of the samples were collected from red wheat, samples from durum and emmer wheat and barley were also analyzed. Our approach to analyzing the samples includes 3 major steps. Isolates are derived from each sample and then race type based on the North American differential set. All the isolates are further characterized on a set of 26 additional SR genes, which are presented in this table, and that includes genes that are effective to UG99, genes important in agriculture, and also genes that we consider universal resistance, where no virulence or marginal virulence has been detected, like for example, SR26. Each isolate is also genotyped with a core SNP assay of 17 diagnostic markers, and a selection of isolates are further genotyped with a custom-made 3K SNP chip. Finally, when races with significant virulences are identified, we use them for germplasm screening. So this graph shows the distribution of races that occur in Ethiopia between 2011 and 2021. The main conclusion we can reach is that the PGT population in Ethiopia has been characterized by rapid shift and replacement of predominant races. But because this is a busy graph, I will present and discuss the result in shorter time periods. Before the 2013 epidemic, TTKSK was the predominant race in Ethiopia. But after 2013, it became almost completely replaced by race TKTTF. Race TKTTF was recovered for about 90% of the collected samples in 2016. The rapid adoption of cultivar Digalu after the 2010 yellow rust epidemic and the large area grown to this cultivar even after the devastating 2013 stem rust epidemic favored the rapid shift of races in this 5-year period. Race TKTTF was first reported in Turkey in 2007 and then detected in the Middle East and also in Germany, the same year that it caused the epidemic in Ethiopia. So this is a great example of how a likely aerial incursion of Viridinium spores from outside of Ethiopia, favored by the presence of a susceptible cultivar, resulted in a rapid shift of races and an epidemic. Now, if we advance 3 more years, the 2016 to 2019 period, you can see that there is another significant race shift. And in this case, race TKTTF was rapidly replaced by races from 2 different clades, race TKKTF in orange in the graph, and TTTF in pink, both from clade 4, and race TTRTF in yellow from clade 3. Also, and although in very low frequency, I want to highlight that starting in 2016, races with virulence on SR24 were detected. In 2016, race TKFTP, a clade 4 race that was also detected 2 years later in Jordan. And in 2017, race TTKST, a variant in the UG99 group. In both cases, these races were recovered from only 1 sample. So the same way we did with TKTTF, we wanted to find an explanation for why races TKKTF and TTRTF rapidly became the predominant races in Ethiopia in this time period. So what do TKKTF and TTRTF have in common with race TKTTF? Well, both races exhibit a broad geographic distribution, including the Caucasus, the Middle East, East Africa, and Europe. For example, TTRTF was the race responsible for the stem rust outbreak in Italy that severely affected durum wheat. And also, all these races has been reported in the Caucasus region 1 to 2 years before being detected in Ethiopia. So these races are also likely aerial incursions from other wheat-growing regions. But when we analyzed their virulence profile, we found that races TKKTF, TTRTF, and also TTTF were all virulent on SR7A, whereas race TKTTF was not. So to assess if SR7A's virulence was responsible for the rapid increase of races, we evaluate the cultivars that are most widely grown in Ethiopia for reaction to races TKTTF, TKKTF, and TTRTF at the seedling stage. And the results are represented in this table. The list of cultivars presented here came from the wheat DNA fingerprinting study published by Hodgson et al. In 2020. As you can see in the table, there is no significant changes in susceptibility when evaluated with the different races. And in particular, in the cultivars that are grown in over 90% of the area in Ethiopia. And SR7A wasn't postulated in any of them. So virulence on SR7A was not the reason for the second race shift starting in 2016. Additional research is needed to answer this question. In the last 3 years in Ethiopia, it has been characterized by a re-emergence of races in the UG99 group, and in particular races with SR24 virulence. Race TTKTT, in dark blue in the graph, first detected in 2019, has been rapidly increasing its frequency to become one of the most predominant races in 2021. Race TTKTT has combined virulence on SR24, SR31, and SRTMP. Another race that is exhibiting a rapid increase is TTKTF, a new clade 4 race. Race TTKTF, presented in light orange in the graph, was detected for the first time in Ethiopia in 2020, and in 1 year became the most predominant race in clade 4. This pie chart shows the frequency distribution of races identified in 2021 in Ethiopia. As you can see, races in clade 4, or the TKTTF group, were obtained from 43% of the analyzed samples. Race TTRTF from clade 3 recovered from about 25% of the samples, and it has been like this in the last 3 years. And finally, races in the UG99 group were detected from 1/3 of the samples collected. The most predominant race is TTKTT. But in 2021, 2 additional races in the UG99 group were detected for the first time in Ethiopia. These 2 UG99 variants are race TTKTT with additional virulence on SR8155B1, an important stem-resistant gene in North American durum wheat that is also present in some bread wheat germplasm, and race TTHTT, a race that is virulent on SR30. Both variants are virulent on SR24, and both were detected in Kenya for the first time in 2019. So inoculum incursion from Kenya might be responsible for the introduction of these new races into Ethiopia. So to assess the vulnerability of Ethiopian germplasm to these variants, we evaluate 106 Ethiopian cultivars and breeding lines for reaction to races TTKTT and TTKTT plus at the seedling stage. As you can see, around 60 and 65% of the germplasm were susceptible to these races. And if we consider only the most widely grown cultivars in Ethiopia, you can see in the table, all but Hidasse were susceptible to both races at the seedling stage, proving the vulnerability of these materials to these races. But I want to highlight cultivar Ogolcho. This cultivar, which carries SR24, is increasing its cultivated area in Ethiopia, and it may be the reason why races virulent on SR24, like TTKTT, are rapidly increasing their frequency in the Ethiopian PGT population. Finally, a few words about race TTKTF. This race was detected from only one sample in 2020, and in 1 year it became the predominant race in clade 4. This race has also been reported in Turkey and Europe, so it is highly likely that represents another aerial incursion into Ethiopia from northern wheat-producing regions. In addition to virulence on SR7A, race TTKTF is also virulent on SR11, a gene effective to races TKTTF and TKKTF. Like we did with SR7A, we will conduct a seedling evaluation of Ethiopian cultivars to confirm if virulence on SR11 is responsible for the rapid increase in the frequency of this race. So in summary, after the 2013 epidemic, the PGT population in Ethiopia has been characterized by rapid shifts and replacement of predominant races. Many of the predominant races has been identified in other geographic regions, suggesting a major role of aerial incursions of uridinospores in Ethiopia's stemras epidemiology. The use of cultivars with narrow genetic basis of stemras resistance, like Digalu and Ogolcho, create a situation of vulnerability and increases the risk of epidemics. Continuous monitoring of the stemras pathogen at both local and regional levels is critical. So in this last slide, I want to acknowledge the technical support and the funding of this research. Thank you very much.